Good afternoon, everybody. Great to see all of you today. It's my great pleasure to introduce our first invited speaker, Nerea Abrego. So Nerea is Associate Professor in Community Ecology at the University of Jyväskylä, right? Jyväskylä in Finland. And her main research interest is to understanding how ecological and evolutionary processes generate variation in species-rich communities, with a particular interest in investigating the scale dependency of assembly processes and assessing the interactions among these processes across space and time. And today's talk will be focusing on global spore sampling project, to uncover the global and spatial dynamics of fungi. So thank you so much for coming to WSL. It's a long journey, we know, and we are very much looking forward to your talk.
Yeah, thank you very much. Thank you very much, and especially for this nice invitation to talk here. So yeah, my name is Nerea, and I work at the University of Jyväskylä. I don't pronounce that well myself because I'm not originally from Finland myself, but I think it's fine for all of us, Jyväskylä. So today I will talk about fungi and about their global spatial distributions and about how to uncover those with spore sampling. So fungi are one of the most diverse, yes, but yet unexplored kingdoms of life. And here we have a figure, a bar plot, where we have like the new species of fungi that are described to science per decade up to 2010. But I can tell you that this pattern is still ongoing, even if we are in 2025. So far, only over 150,000 species of fungi have been described, but the estimates of the global species richness, they vary from 0.5 million to 10 million species. And why is this? Well, there are like many reasons, like what is the traditions in different fields of ecology of studying one group or another group. But one big reason why fungi still remain so little known to science is that most of the knowledge that we have of fungi is from those fungi that produce visible structures like the fruiting bodies of macrofungi, but most fungi, they don't produce such structures. They remain microscopic their entire life cycle, or they may sporadically produce a fruiting body, but is like for a very ephemeral time period. So this poses a challenge for us wanting to understand what are the distributional patterns of fungi, like if we rely on morphological surveys. The environmental DNA surveying techniques, such as taking an environmental sample and applying metabarcoding, is totally revolutionizing the fungal research because with these new techniques, we can target these species that they have remained hidden for us. So for example, here we have a cartoon of a log where few species are fruiting, and those are the ones that we could detect with fruit body surveys, but there is much more going on within the log. And with eDNA surveys, we can target those and know more about fungi. But of course, like when applying eDNA inventories to study fungi, we additionally have the challenge that fungi are very substrate. Fungal communities are quite substrate specific. Different substrates, soil, wood, living plants, they hold very different fungal communities. So if we want to go to one forest site and comprehensively survey fungi applying eDNA, substrate-specific eDNA surveying, it can be quite costly because we need to target many different types of substrates, collect lots of samples, and that costs. And of course, we need to identify the species molecularly, which is another challenge, but we'll talk about that a bit later. So this is where the Global Spore Sampling Project comes. It's a project where we try to understand the global diversity patterns of fungi by collecting spores from the air and identifying those species. And before I go into explaining that project, I want to give you like a little bit of a background on the history on how we ended up with this idea. So back in 2018, I met with Vera Norros, who had done her PhD on fungal dispersal, and she had collected with these devices, the cyclone, which we call the cyclone samplers. She had collected some samples with those, and she had already done the DNA extraction and sequencing and all. Her idea was to use them to understand about fungal dispersal, but she was not like very sure on how to use them, and she contacted me and Nerea, what could I do with this? And I was like, I don't know, let's have a look to the data. Let's see what is there in the air. Like, so these data were quite small. They were from 4 sites in Finland. And just looking at the species composition from the data from this device, we saw that the spore fungal communities, there were some patterns in time, like in different weeks of the year, we would find like different species in the air, which we knew from the phenology of the fruiting fungi that we have like different species through the season. But more interestingly, we saw like some patterns of the different sites in Finland. They have like quite different fungal community compositions, and this pattern was not just random. Like there was like some structure in the data, and so it occurred to us that can it be that this method actually is like really good to understand like what fungal species are in different places. So then what we did to understand this a little bit more was to, so this Venn diagram here is from one of these 4 sites, which is Kuusimäki, which is a site that has been like very intensively surveyed for fungi. So we knew very well the fungal communities that were in that forest, and we compared to the species list that we got from the air. So we compared the both substrate specific species list, the soil specific species list, we compare to what we found in the air. And to our pleasure, like what we found was that there was like quite a big overlap in what we found in the air versus what it was known before from that site. So you can see here, like this overlap here is pretty large. And additionally, we knew about many more species. So from this, we started to think, okay, this method seems to be informative about fungal species composition in a way that we have not been able to know about fungi before. So to test a little bit more this method, we did one study where we did this spore sampling in 5 cities in Finland, where we sampled the air communities from the urban core areas towards their edge with the natural, like the native forests, and towards the more deep in the forest. And what we wanted to understand was like the effects of urbanization on fungal communities, but also from a methodological perspective, what we wanted to understand is, can we capture the differences in fungal communities that urbanization is posing based on data from the cyclone samplers, from the spore air sampling. And I mean, in my mind, I was thinking like, well, possibly we will get to know like the differences from the urban core to the natural core, but I am pretty sure that here in this 1 kilometer distance from the natural edge to the urban edge, we are not able to detect the differences because it's just 1 kilometer, and what is in the air, in my mind, it was like, it's mixing. But to my surprise, we found a huge difference in the species diversity and community composition already here. So then, I mean, I got super excited, and I discussing with Tuomas and with Otso, we started to think big, and we thought like, we have to do a global study. Now we have the tools to finally get to know what is going on with fungi, and this is how the global spore sampling project started. So then we contacted several, as many groups as possible around the world who would be able to do like weekly sampling with these devices, weekly 24-hour sampling with these devices. And the end result of the data collection is like this. We got quite a nice representation of the polar continental sites. Of course, we have most places are from Europe and from Central Europe. Even in Zurich, we had 2 places. And we have the temporal coverage of over 1 year. Of course, like the tropical sites, they are the most scarce in the data, but still we were quite satisfied with this end result of the sampling representation. The bioinformatics were pretty challenging. It's not something that is like my core expertise, but I'm with Brendan, who was a postdoc working with this data. He developed a species assignment method that is probabilistic, so that the challenge that we have with fungi is that many species are yet undescribed to science, so it's really difficult to assign the species name to the sequences, and there are a lot of biases in the reference databases, like some branches, some clades are completely unknown, whereas others are better known. We have biases in the spatial representation of the sequences, but he developed this method with which we were really happy that we could classify the species as known taxa, as totally novel, like known taxa, so that we could assign a name, novel taxa that we cannot assign a name, but we are pretty sure is a species, and known or novel taxa, so those for which we are unsure. And the data comprises around 24,000 species level OTUs. And what we wanted to answer with this data are like actually very basic questions in biogeography. So they are things that have long been known for animals other than arthropods or plants. So how do fungal communities differ among the major bioclimatic zones, and how much the climatic variables explain such differences? How do the global seasonal patterns in airborne fungi vary with latitudes and weather conditions? And are the responses of fungi to climatic and seasonal factors phylogenetically and functionally structured? So these were the questions. To answer the first question, do we have do the major bioclimatic zones structure fungal communities? The answer is a very, very clear yes. Our data showed that, and especially the mean annual temperature is a major driver of the fungal compositions across the world. We also tested for many other variables, but climatic variables, but this mean annual temperature is clearly the strongest predictor. More interestingly, how do the global seasonal patterns of airborne fungi vary with latitude and weather conditions? And here, I mean, you can see there is a lot of noise in the data, but generally, what we found was that as we increase the distance from the equator, there is much more seasonality in the fungal occurrences in the air. So if we go more to the tropics, there is little variation in the amount of species we find in the air, the DNA amount, and also there is less variation in the species turnover over time towards the tropics, whereas the contrary is true towards the poles. So when we go more to the Arctic, we have like this very strong seasonal dynamics that when the winter comes, basically nothing is there. The growing season comes, and then we get a lot more species. Many people have thought that previously that in temperate climates, we can even exceed the species number of fungi than what we have in the tropics, but our results show that no, that is not the case. Like fungi are following like this type of very typical species richness patterns that have been known for other organisms for long. We also did like some more deep modeling that I will not go into detail, but up here we have like the species that we had in the data, their taxonomic tree, and here we have like the responses for seasonal and climatic variables. And what we can see here is like many different colors, which just means that there is a lot of variation in how different species respond to these climatic and seasonal variables. But the take-home message here is that if we look at the seasonality of species, these responses are very taxonomically or phylogenetically conserved across fungi, so that groups of phylogenetically related species, they show similar seasonal dynamics. Let's say those species that occur mostly in the autumn, those that we like to pick up for eating. So there is like a very big phylogenetic signal in those species belonging to the same group. But that is not the case for the responses to other climatic variables like weather conditions. So that has been broken for fungi like through their phylogeny. So so it seems that different fungal species around the globe, they have been adapting to the local climatic conditions independent of their phylogeny. We also looked at functionally, or well, regarding the traits of the species that we found, whether there were like any patterns. So we did not see the spores in the samples, but we could assign by assuming that the species were in their asexual life cycle or sexual life cycle, we assigned the spore size, and what we saw was that the asexual spore size decreases, but the sexual spore size increases with increasing the distance from the equator. And I will now explain that a little bit. But if I need to conclude these results, I think that these results, what they show is that fungi have like very predictable patterns of spatial and seasonal variation, and that these results really suggest that the drivers of microbial community assembly, of fungal microbial community assembly, are very similar to those determining the assembly of macroorganisms, which was not known so far. The results suggest that fungi have continuously adapted to the climatic conditions rather than being stuck in their ancestral climatic niches. And regarding the spore size of sexual or asexual life cycles, it seems that there are contrasting evolutionary forces behind the size of sexual and asexual fungal dispersal propagules. I was not alone for this work. We had a lot of collaborators who actually did like very hard work because the sampling was very intense, once per week for over 1 year. And I cannot finish this talk without very deeply thanking them and to you for listening, but they did more work. Thank you.
Thank you so much for your talk. Is there any question in the audience?
Thank you. I was wondering, when you compared urban and the natural areas, is there a different wind structure that may play a role there? Like maybe if I would assume that maybe the built structures prevent like wind, and then you would maybe find less fungi species in the air because of this.
Yeah. I think when I talk about urbanization, like I'm talking about like all the cumulative effects of having these urban buildings. So of course, like that is one of the things, like maybe spores cannot move through the buildings. Yeah, I don't know if I answered the question.
So this may not represent the diversity itself, but rather like the ability to reach a certain point, right? So I'm just wondering, like when you sample at some location, how large you assume the area of where the spores come from?
Yeah, this is the question. Yeah, I don't assume that, because it may depend very much on the landscape characteristics. When I did my first study back in 2018, I was convinced, and I think in the article I even gave the number that more or less what this air sampling represents is like something of what is around in 100 kilometers. I thought that, but I think I was wrong, because then I have done like more and more studies at a smaller scale with some ecological contrasts at the small scales, like the one I showed, but also others. And I am seeing that this spore sampling can inform about the changes in fungal diversity at very small scales, like a couple of kilometers. I don't know exactly what is the area of representation definitely is larger than if you sample like soil directly, because that is very local, but something larger, but I cannot say because there are many other factors that influence this. Like, is there like a strong wind? Are we talking about an open area or a forest? And so on.
Thank you.
I have a question to the plot that you showed of the seasonality of species richness. I think it was, yeah, this one for the different climate zones. It seems you have quite a strong line at zero here. Is this an effect of weather conditions, or how do you explain this?
Well, if you look at the color that is dominating that zero is purple. That is the polar continental place. So it's like, I mean, especially in winter, those even if we collected the spore samples in the winter, there is nothing there. Everything is frozen, and generally there is also like a lot less diversity up there. So also like the DNA extraction and everything becomes like much more tricky from these samples when there is so little DNA. But yeah, if you see like is mostly the purple, okay. The polar continental.
Thank you.
Not. So then I have a question. Was it possible to relate the amount of spores retrieved by your trap system or the amount of DNA you could extract from them and the density of potential hosts, at least for a few selected fungal species?
Yeah, unfortunately for this study and when doing this DNA metabarcoding, we need to destroy the samples to extract the DNA. And the spores are so small that we could not count them before extracting the DNA. We have thought of many ways in which we could possibly do that, like how to count the spores and also like how to measure them, like the real sizes, but we haven't come up with any solution that wouldn't compromise the sample for the DNA analysis.
Yeah, still some work to do.
Yeah, like we have really been thinking about this. I'm sure somebody will come up with something, but we haven't like really cracked the system yet.
Thank you. There is one last question.
Hey, thanks for the talk. It was really nice. I was just wondering here in the graph, looking at that, how did you deal with data from the northern and southern hemisphere? Because I guess this is more from a northern hemisphere perspective. If in the middle it goes up, did you like lump it together, or did you somehow change the day of the year, or just to understand the graph?
I don't know if I understand.
Because the day of the year is like in the middle, it would be, for example, here.
Like with the different hemispheres.
Yes, yes.
Yeah. We trick the data for that, of course. Yeah. Now. Yeah.
Because otherwise.
Yeah. Otherwise, like we would have. Yeah.
Okay.
Yeah.
Okay. Interesting. Thank you.
Yeah, this is just for simplification. We show them like this, but of course.
All right. Thank you again for coming to WSL, and we go now to the next talk. Thanks.
Thank you.
Hello, everybody. Also from my side, it's my pleasure to introduce the second speaker of today, Fabian Schneider from the University of Aarhus in Denmark. So Fabian began his scientific journey here in Switzerland. So he made his PhD at the University of Zurich And then he did a postdoc in California in the Carbon Cycle and Ecosystem Group at the NASA Jet Propulsion Laboratory, where he then also got a research scientist position in 2021. And after that, at this time, he then developed new methods for monitoring ecosystem and biodiversity change with remote sensing from space, which is also his big interest in combining remote and in situ observations to then understand how climate and biodiversity changes are impacting ecosystems and their functions. And since a bit more than a year, he's now professor at the University of Aarhus in the Ecoinformatics and Biodiversity section, and is also having the lead for the digital landscape analysis at the Landcraft Pioneer Center. And today he will give us a talk with the title Biologists, the Field and Satellites in the Sky. So are we in a digital revolution for biodiversity monitoring? I think this will be very interesting for lots of us. And I'm very much looking forward that you give us this talk. And thanks again also for making the way to Wilmersdorf to the WSL. So the floor is yours. Thank you.
Yeah, thank you very much. Thank you very much for the introduction and also for inviting me here. It's really exciting to be here. And as you see from the title, I'm really trying to integrate what we do on the ground in the field with remote sensing observations from the air and from the sky. And hopefully at the end of this seminar, you also get a little bit of insights, whether you can answer for yourselves, whether we are actually in a digital revolution in how we monitor biodiversity. But first, a little bit of background. So probably we scientists in this room know that we're losing biodiversity. But I think it's important also to see that if you look at the World Economic Forum Global Risk Assessment, and we look at these assessments of the past couple of years, then we see that the perceived risks for society of the following decade are all environmental, here shown in green. Over the years, the terminology have changed a little bit. For example, the term climate change is not here anymore, but we see that the number one perceived risk is extreme weather events. And number two is biodiversity loss. And also in this new report, they include the term ecosystem collapse. So I think it's important also to see that we're losing species, but we also see ecosystem changes like degradation of ecosystems or loss of functions. So what can we do about it? I come from a remote sensing point of view, and what we try to contribute basically is to better understand where, what, and how are things changing in terms of ecosystem changes. So we need to monitor to understand and act on biodiversity change. And there's a lot of new possibilities how we can do that. So there's a whole range of new Earth observing satellites that are being prepared and launched in the future. This figure here shows an overview of from ESA about new so-called Copernicus Sentinel expansion missions. So these are all new technologies and missions in development. And I want to highlight two types of instruments here. One, it's called imaging spectroscopy. So it's a new group of spectral instruments that look at how sunlight or radiation is reflected from the Earth surface in many, many spectral bands. So you get these spectral signatures of the Earth surface that can tell us something about surface properties. So if you look at plants, it can tell us something about leaf chlorophyll or leaf water content. And we have a global mission from ESA called CHIME that is in preparation, that might launch in maybe 5-6 years. But we have now some new satellite data that is already available, like the German MMAP mission or the Italian PRISMA mission that provides like snapshot images of Earth 30 by 30 kilometers from this data. And that allows us now to test what we could do in the future if this kind of measurement becomes global. The second part is looking at vegetation structure or surface structure with so-called active remote sensing. So that includes LiDAR and radar measurements. So in terms of LiDAR, that's basically sending laser pulses down to Earth. In this case, the JEDI mission is doing that from the International Space Station. And then you measure the reflected energy that is being reflected throughout the canopy and from the ground. And from this information, you can derive information about canopy structure of forests. And you can do similar things with radar instruments. So what are the areas where we think we can contribute with this when it comes to what's called the biodiversity plan that came out of the global biodiversity framework? So that's a policy instrument that defines goals and targets for the next decade. And in terms of these targets for 2030, it's primarily the ones that link to area-based conservation, where I think we can contribute a lot with remote sensing, like plan and manage areas, restore 30% of area or conserve 30% of area. So maybe some of you have heard of the 30 by 30 target to protect and restore 30% of area by 2030. And then there's other areas like mapping invasive species or looking at resilience of ecosystems to climate change, sustainable agriculture or fisheries, and nature contributions to people or urban biodiversity, where remote sensing can play a role, but it's maybe not the major contribution. And finally, I think the space agencies also contribute quite a lot to open science, because all of the satellite data, at least from ESA and NASA, is openly available, and they also invest into making this data more accessible. So kind of the aspects that we can cover here is like, where should we protect? Where is high or low diversity? What is there? What kind of land cover type or plant functional type do we have? And so that's more of the spatial aspect we get from remote sensing. But where we really want to go, I guess, would be the how. And this is where still the developing areas lie, basically, to find out, for example, using remote sensing, how effective are conservation or restoration measures, or how are we doing in terms of like bending the curve of biodiversity loss. So in this talk, I want to give two examples from my research, from some of these, like, let's say, newer technologies that are at least new in space. And one is this LiDAR instrument that is sending laser pulses to the ground. So then you can imagine there's a laser pulse, it's 25-meter wide, that hits the canopy and reflects laser energy through the canopy, and you get this vertical profiles, these waveforms that tell us something about canopy height and structure. And then the second example is using this imaging spectroscopy technology, and to link it with sampling on the ground, where we measure, for example, we sample leaves, measure leaf traits, and link that to the spectral signal to be able to then map different properties of the Earth's surface. So the first example is trying to monitor the structural diversity of forests from space using this JEDI instrument. And basically what happens if you put that on a space station is that the space station is like moving around Earth in going north and south between 52 degrees north and south. And you get this kind of scan patterns of laser pulses. And if you integrate that over time, so here it's about 3 years of data we integrate, we start to get a dense enough coverage of laser shots, here illustrated in the Western US, around California, that we have enough of these laser pulses that we can actually look at vegetation, structure, and structural diversity. So this is what we did, trying to map this as a property of ecosystems, this structural diversity in the Western US. And yeah, you see this bright yellow areas where we find the high structural diversity, and then lower diversity in this darker colors. And basically how we derive this from the JEDI instrument is that we looked at 3 properties of the canopy. First is the canopy height. The second one is the vegetation cover, so a metric of density, both density of leaves in the canopy, but also density of stems in an ecosystem. And then finally, the foliage height diversity as a metric that relates to the vertical heterogeneity of the canopy. So whether you have layering, whether you have understory and so on. So then we combine this 3 metric into a multi-dimensional space. We could also call this a trait space or a feature space. So we use these traits as axes, and then we map these laser shots in an area like a square kilometer here in this 3D space. And then the metric of diversity is basically how much of that volume or how much of that niche space is covered. So, for example, high diversity would mean maybe we have many different species, many different age classes, and a lot of coverage in that functional or structural space. Whereas low diversity could mean maybe adaptation to very specific conditions. It could mean a monoculture, or that only a small range of that space is covered. And because it's a multidimensional metric, it also captures things like how correlated are actually some of these metrics. So you could have, if you look at them individually, they might have the same range of variability, but together you might still see differences in how much of that niche space is covered based on how correlated the properties are. And to give one example, here where we find particularly high structural diversity is here in the Sequoia National Park, where we have the sequoia trees, some of the largest trees on Earth in terms of biomass, and also very tall trees, 80 to 90 meter tall. But it's an ecosystem that has frequent disturbances, especially fire disturbance. So you have a very, very heterogeneous canopy, a lot of height variability, but also a lot of different species and heterogeneity in terms of forest structure. So then the second example that I would like to give relates to monitoring plant functional diversity from space. And to test that, we use here airborne imaging spectrometer data from the Devis instrument. So they were flying particularly high altitude to simulate data, how it would look like from space. So we can start to test and build up our methods. And basically what we're getting from this hyperspectral signal are these, oops, this is a little bit off. I think I can show you like this. So on the left part of the graph, you're in the visible part of the spectrum. You have a lot of absorption of radiation from chlorophyll and other pigments. Then if you move into the near infrared part of the spectrum, you have a lot of scattering. So it links to leaf structure, to vegetation structure, but also water content in the leaves. And then if you go into the shortwave infrared, you have a lot of water absorption. And on top of that, you have like some interactions and absorption features related to proteins, lignin, and cellulose. And that's why you can also map things like leaf nitrogen quite well with these kind of instruments. So this is showing where we are and the coverage of this kind of, let's say, test data. This has been acquired in California. So maybe some of you know the Bay Area, and now it works again. So San Francisco is here, Los Angeles is down here, but we are actually looking into the Sierra Nevada mountains here, where we have 2 large flight boxes around Yosemite National Park and around Lake Tahoe. So the example I would like to show here is for one trait first, that's the leaf mass per area that we were able to map across this region at 30 meter spatial resolution. So that's about what the satellite data is going to look like. And you see some pretty strong gradients here in terms of leaf mass per area. So that links to the strong elevation gradient that we have here. So if you go kind of from west to east, you have this kind of more kind of oak woodland savanna in lower elevations and oak woodlands. Then you get into kind of this lower and upper montane forests. So that mid-range is also where the sequoia trees sit. And then if you get to the very high elevations, you have actually pine trees that grow up to 3,000 or more than 3,000 meters, maybe almost 3,500 meters. And you also have a lot of this kind of subalpine, alpine vegetation, including meadows and a very steep eastern slope. So what we were doing is we were actually mapping a whole set of traits, like 8 different plant traits. And then for this visualization, we distill this information into principal components. So then we visualize just 3 of these components. It's a dimensionality reduction. And then that's how we map the colors here. So you see basically the different colors here represent different groups of, you could say, plant functional types in a way. That's how the traits group together. And you also see the strong elevation gradients. And then we see how these individual traits map onto these principal components. So it's a little bit messy, but I want to highlight here 2 main axes of variation that we see. One is very well known. That's a kind of a trade-off between plants that have higher leaf nitrogen, invest more into productivity, and then plants that invest more into higher leaf mass per area or higher longevity of leaves, and also show maybe slower growth rates. So some people also describe this as the fast versus slow growing like species range. So we see the same axis as we see also in global trait data. And then we have another axis that we are still trying to understand, that it seems to link a little bit to like structural versus chemical. You could say maybe it's structural defense versus chemical defense. We don't fully know yet. But on one side, we have more plants with higher lignin and also generally taller trees. And on the chemical side of things, we have plants with higher leaf phenolics. It's also one of the traits that we could map. But I'm particularly interested in diversity patterns. So we also had a look at what happens if you now, for each square kilometer, look at the range of traits of each individual trait and whether these individual traits actually show kind of the same patterns of diversity across the landscape. And we see that a lot of them actually do group together. So we have this yellow area here with very high variability in the traits that a lot of them share. But interestingly, we also see a second cluster, and that's nitrogen, LMA, and starch that show actually the highest variability at higher elevation. And then we also see that chlorophyll is kind of on its own here, and it shows the highest variation in the lower elevations where we have the oak woodlands. So we are now investigating what does that mean if we then map these functional diversity patterns in multiple dimensions. So what happens if we go from single traits, 1D, into many traits and multi-dimensional trait spaces? And one thing we see that happens is that some of these diversity hotspots get more pronounced if you go into more dimensions. That makes sense in the sense that it's much harder to fill a multi-dimensional trait space. So there's much fewer places where you actually have such a big variation of traits that you could have reach a high diversity in a multi-dimensional space. Maybe you also see that it starts to look quite similar. So we generally say maybe you need maybe 3 to 5 different traits to capture a lot of the variation in trait spaces, at least that we can measure with this technology, because there's also a lot of correlation among the traits. But I wanted to give one example here that's maybe a bit simpler to grasp. That's looking at the 3-dimensional case. And what we actually show here is we tested all different combinations of traits together and then looked at the mean pattern. And then I also looked at which combination of traits ends up to be more similar to the mean. And you kind of see that like corresponds to the previous slide, that there's this group of traits, they all seem to show high variability here in this like mid-elevation range and also here. And we are still investigating what that means, what could be the driving factors. One thing we know is that this northern area has much more forest management, and there's some really, really strong forest management patterns, including large clearances. And that influences these large scale patterns. And then we have some very like heterogeneous environments like the Yosemite National Park with very complex topography that also leads to high diversity. And finally, we looked at which trait combination would be most different from that. And that also maybe reminds you from what we've seen before, that leaf mass per area and nitrogen and starch show really high diversity in very high elevation. And this seems to be primarily linked to the heterogeneous mixture of plant functional types, including grasslands, shrublands, and forests. So I'm running a little bit out of time, so I try to speed up for the last part, where we basically just want to give a little bit of an overview. How do some of these remote sensing methods map onto essential biodiversity variables and indicators? And to sum up, basically, we put satellite technology on the right and type of indicators on the left. And we see that we think that these future technologies can primarily tell us about ecosystem changes and also drivers of change. So very, very good in mapping disturbances, for example. And then we have a lot of also ground technology that is evolving. Ecological monitoring gets kind of more digital, using microphones, using digital cameras like camera traps, and using eDNA sampling. And we did a bit of like a literature review, and then also like an expert assessment, how we would like assess all these different technologies in terms of their potential for automated or semi-automated biodiversity monitoring across different realms like freshwater, marine, and terrestrial. And we kind of grouped it by what we think, what is like the readiness of some of these technologies, and how many different essential biodiversity variables they could inform. So that shows us kind of where the potential lies. And we think that this passive remote sensing techniques on the ground and from space show some of the higher readiness to be deployed in a more automated way, but that actually eDNA has like the biggest potential to inform very, very broad taxonomic ranges, but it might not be ready. It's not so easy to automate it, but it has a very big potential for development. And yeah, like to give the last example is basically the biggest potential we see is if we start to integrate these technologies. So to integrate that, we looked at the large network of bioacoustic sensors that can be used to detect bird species. And we linked it with a large geospatial data set that connects abiotic and biotic variables derived from remote sensing. And we looked at the how birds basically use a niche space and like geographic ranges. And one thing we saw by doing that I think is very interesting, that when we look at different genuses, we kind of see, of course, patterns of more specialists, more generalists. Some have a smaller habitat space they use, and maybe also smaller geographic space, and they maybe don't overlap much. So we see like a positive relationship. But if we looked at birds from the same genus, we saw an interesting pattern of like niche partitioning maybe. So we saw, for example, birds of the same genus that use the same geographic range seem to very well partition the habitat space locally. And then birds that occupy the same habitat space really partition their geographic space. So that's one of the new kind of macroecological findings that we can get if we combine these different technologies. So to wrap up, are we in a digital revolution for biodiversity monitoring? I mean, there's a huge range of new satellite data coming up. There is also a huge increase in data we collect, for example, in the Global Biodiversity Information Facility like GBIF. Basically, the data sets are exploding, you could say, and we can look at global databases like the Tri-Plant Trait database that is continuously increasing in terms of the data sets that are there. Or another example is animal movement data. So here we have a log scale. So basically there is orders of magnitude more animal movement data and also more automated data available globally. So it really opens a lot of possibilities for integration. So yeah, my final point is that all these technologies alone maybe don't give us much, same for the satellite data, but we have to think about integrating it with our data on the ground and with models and experiments. Yeah, so with that, I really like to thank you for your attention.
Yeah, thanks a lot, Fabian, for this very interesting talk. I am sure there are some questions.
Thank you for the talk. I think it's exciting to see what's coming, what we'll be able to access on a more global scale in coming years. I'm curious with the functional representation of the landscape that you're looking at. A lot of that is likely driven by, I mean, we know it's driven by species differences across these gradients and stuff. One way to go at this, or one way that we've studied functional representation, is to look at species and then go through databases like TRI and then map functional diversity. You are going at it the other direction. I'm curious what additional information we gain by looking directly at the functional aspect, because there's something we gain from looking at species, there's something we gain from looking at functional bits. And so how do we, have you looked at both of those together with the perhaps joint SDMs of the plant species distributions in this range?
We have looked at it a little bit because, of course, there's a big interest in understanding how this functional and taxonomic diversity relate to each other. I think one additional thing that we are seeing, especially with very dominant species, like if you take European beech, is that we can, of course, see intraspecific variation, and we see a lot of variability based on the environments the plants are growing. But it's often quite challenging, actually, to bring the two together, because it's a matter of scale. And I think that's why it's so exciting to have maybe now camera trap networks, where we get animal occurrence data or distributions across large spatial scales that we can connect, for example, with functional trait or diversity maps to see how habitat space and animals link to each other. Or we can use the bioacoustic data also at scales that make it more possible to connect the two. So I see a lot of potential to do it. I haven't fully integrated it because we also have to see that we only see the surface most of the time, so more dominant canopies. So it would also be very interesting to learn what can this functional composition of just the dominant canopy tell us about the whole ecosystem, because we are basically neglecting the understory most of the time.
There's another question here.
Hi, Fabian. Thanks for this talk. My name is Fabian too. We do ground truthing through airborne environmental DNA. And we're also part of ESA-BIC, which is very much this idea of connecting satellite data and ground truth biodiversity data for biodiversity prediction. And so this is super interesting, and I have two questions. The one is a question of scale. So how most biodiversity monitoring questions, especially if you talk about industrial impact, is very small scale, right? It's about this field versus that field. It's about how do you link satellite data down to these very small scales, because most studies I think are done at scales, continental scales, scales of regions. Do you get patterns at very small scales too that are informative? And the other question, which is a bit erratic, but in the species distribution modeling world, it seems to be that the addition of satellite data on top of the classical predictor variables like the bioclimatic variables don't necessarily give you much more predictive power. But that might be different studies, different type of satellite data. But have you looked at that? What more predictive power do you get from using these advanced satellite analysis data? Thanks.
Yeah, so to answer your second question first, or I start addressing because Devis will talk afterwards. So maybe he has more to say about species distribution modeling. But one thing we see with the data we have now seen in the Sierra Nevada mountains in terms of like mapping like bird species habitats or distributions is that I think it's also like a matter of scale. So if your ground data is only available every couple of kilometers, and maybe you capture also a lot of, for example, generalist species, then of course a lot of your patterns will be explained by climate gradients, and it explains almost everything. So maybe then some of these new variables don't give you so much additional information. But I feel like if you look locally, and that's maybe also the difference there between like geographic range versus kind of local habitat space, I think we should have a lot of additional like information about the habitat use in local places of species. So I see a lot of potential. I haven't fully tested it, like how much additionally it actually tells us. And that's the question about scale. It's true that a lot of diversity monitoring and also action management is local. So something we do for testing how well it scales up to space is to use like intermediate remote sensing measurements like airborne, or now drones become quite available, to have some sort of upscaling mechanism to maybe go from individual leaves, plants to actually communities, and then from the communities to the space. But it might not be possible that we ever predict like alpha diversity from space, but maybe we predict beta diversity or species turnover quite well, for example.
We maybe have two quick last questions. One was here and one was over there, and then we move on with the program. You can ask later. Okay, then we have one last question, and then all the others can ask later during the apéro.
Okay, wow. Thank you very much for your talk. I'm always in awe when I hear remote sensors talking about everything that is possible these days with remote sensing data. And it's really nice to see from your perspective that we can also test a lot of things and use these data to confirm some of the ecological patterns that we have discovered perhaps or acknowledged in the past. So my question is really, what do you think that is for ecologists and for biodiversity research that use remote sensing? What do you think is out there that we can learn from these data that we don't yet realize that we could know?
Yeah, that's an interesting question because I don't know what you know, but I think there is just one thing to say because there's such a big focus on like NDVI as a metric of greenness. And of course, it's very powerful if you look at phenology. And I think that's where most of the information content also comes from, like differentiating different communities by their phenological signal. But just that to be aware that there's a lot of other possibilities now to look at structural aspects of plants, for example, or biochemical or biophysical properties that become more and more available as well.
Okay, thanks a lot. Thanks again, Fabian, for coming to WSL. And a big applause for you.
Thank you very much.
And I hand over to Nick.
Thanks again. So our last speaker is Devis Tuia, and he has completed his PhD at the University of Lausanne on kernel methods in hyperspectral satellite data. So it's a bit similar to what we have just seen from Fabian. Then he moved on, did a world tour as a postdoc, went to Valencia first, University of Valencia, then to the Colorado University in Boulder, Colorado, and came back to EPFL, where he did an additional postdoc, and then he moved to Zurich, became an assistant professor at the university, then moved to Wageningen, became the chair of the Geoinformation Science and Remote Sensing Lab, and finally, since 2020, he is back to Switzerland in Sion, EPFL, where he is the head of the Environmental Computational Sciences and Earth Observation Laboratory. Today, he focuses on Earth science from above with machine learning and computer vision. And he gives us a talk on deep SDMs, modeling species distributions across scales and data modalities. Please, Devis, welcome.
Thank you very much, Nick. How are you doing? Yeah? Oh, good. Great, great. So we have already spoken about very interesting things. We've heard about the life of fungi. We heard about sensors. The last controversial topic we haven't really touched upon, it's AI. Yeah, I know. AI, that's what I'm going to talk about. So I hope there is not too much skepticism in the room. I'll do my best to show you a little bit the pros and cons and see how we can put this to the service of biodiversity monitoring. In my case, I'm going to talk about species distribution models. And so I will talk about two things in particular, two works. And it's obviously not only my work, but it represents the work of a whole group. And I hope you will enjoy it. If not, 20 minutes, then the apero, it's also fine. Okay, so I didn't know you will show the the graphics, so you get it twice. Does it work? Yeah, I can point here, and it also works. Amazing technology. So I mean, I suppose I don't have to convince any of you about the importance of changing these curves into something more sustainable, because we are not doing a very good job at it at the moment. And I know it's always easy to talk on a public of convinced people. We know that we have to do something. And today, at least, we have seen different ingredients, no? Community sampling, sensor, and now I'll show you a little bit of what we can do with modeling, okay? Because if we do nothing, then we lose a lot of species and populations, and this is definitely not what we want to achieve. Because all these all species give us a lot. It's not just aesthetic values, but it's also medicines, food. I mean, again, I don't have to convince you about all this. Then okay, it's not, the order is up to you. Okay, then let's not start a debate whether climate change is bigger than biodiversity collapse. They're all extremely annoying for us and for the planet as a whole. Okay, so the thing that we want to do is effective conservation. Okay, we need to at least provide science-based evidence of what's going on so that we can support people who are taking the decision so that they take the right decision. I'm no politician. I have no real power over conservation strategies, but the thing I can do, well, it's modeling. So the thing I want to provide is good evidence so that the decision makers then can make the right call. And maybe I need the list in that point, but I still think that this is possible. So if we want to have effective conservation, we need to map, and we need to map well. So that's my hypothesis for today, and I hope, I guess many of you in the room more or less agree, at least to a little extent. Now, the idea here is to try to go big in a way, to map as much as we can, as big as we can, with all this wonderful sensor data that Fabian has talked about, and all this wonderful community-based sampling exercise. And if we put all this together, maybe we can do something that can be used. Okay, but I still haven't said the magic word. I still haven't said the two letters word, that the AI one. Oh, I just did. And so I just wanted to, I'll do my test like the others. It is delayed. Yeah. So all the images you will see here have been very graciously generated by DALL-E 3, which is a generative AI model. So I prompted some of how I wanted it to look like, and it's a little bit to see the thing that we can do nowadays with this type of technology. Of course, there's nothing scientific in that, but just to show you that the level of image generations that we have nowadays is quite impressive. I could generate the same scene in photorealistic terms, and I think most of us would just believe it's a photo that I've taken while walking. So, I mean, it has advanced a lot, and it's something we need to acknowledge. Now, the question is, how can we use all this technology for the well-being of our planet? And I will use as a first starting point a work I couldn't resist that I did with Nick, and that you can see there. And it was a little bit our first entry points in species distribution models, and it has been a love at first sight between me and SDMs. Okay, but before showing pictures and models, I have a question for you. I mean, coming to WSL, I expect you are prepared and you can answer this question. So how many species of trees are there on Earth? 58,000. 80,000. Do we have more, less? I mean, it's pretty good. When I gave that talk to a computer science public, they say 100. I'm not making that up. It's true. So 73,000. So Nick was very close. And so I didn't know myself first. I had to look it up. And so we know that there are more or less this number of species. Now, the second question is, where are they? And this is, in a way, the question that we were trying to answer with species distribution models, okay? Given the climatic conditions that we can observe, can we say if a place is suitable for a species or not? The story is that the world is big, okay? And we don't know exactly where all these species are. If we want to be like global precise, if we want for every one of these 73,000 species, knowing at every location on Earth if they're suitable or not, well, we have still a lot of work to do. So this is like kind of a first element of response to that question, okay? Now, the next question that I'm interested in is like to see where they will be, like where we know how to make climatic projections to the future. So are we able to take what we know of today's situation and try to make an educated guess of what this distribution will look like in, I don't know, 100 years from now? Okay, you all know a lot about it, but there is a lot of uncertainty. You have all looked at the IPCC reports. Uncertainty goes like very, very, very big at some point. So we do it the best we can with this type of estimate, and we will see where this will lead us. Now, a little bit of AI. So I don't know if you're familiar with machine learning as a discipline in general, but basically the thing that you try to do, so I hope that behind me a pointer is appearing. Oh, it's horrible not to know. I will turn, sorry. So you have some inputs, so that we call some environmental covariates. Then we have some observations that are here. Oh my God. And then we try to relate inputs and outputs. So the values of climate, soil, you name it, and a 0-1 number, whether the species is there or not, through a certain model that will basically look at an input, try to predict the output. Am I right or not? If I'm not right, I change the model. I do it again. I do it again. I do it 1 million, 2 million, a gazillion times. And at some point, the model starts to get it right. Okay? So this is like a very, very crash course. Machine learning is basically example-based learning when you learn the inner functioning of a model by observing inputs and outputs over and over and over again. So you can see that this is very, very different from mechanistic models, but it's just another approach, and it's very complementary. So these are not two words that should live apart, but that's not the topic of today. So the topic of today is what we did. So we started with these environmental covariates, the observation of 10,000 species. I am unable to go up there. Yeah, here. It is very difficult. And why 10,000 species? Because we had these 13 databases, and we took all the species that had at least 20 samples, if I remember well. So this reduced from the 30,000 we had to 10,000. there is no record for 73,000 species in the databases that we used. Okay, and then we started using some very traditional machine learning models. It's some random forest. So this is a model that will pick the variables, try to split the data, do it again, do it again, do it again until you get it right. And then some another type of decision trees. We ran many of them and we took an average, because the average is always good, because it kind of erases the errors of the single models. So that the majority wins. And this is particularly good for species when you have just a little observation. Okay, good. So thanks to this, we could bring a first answer about where are they. Okay, we could run this using all the climatic variables from Chelsea. By the way, today I met the maker of Chelsea. So are you here? Oh, sorry, I should not call people out. This is very bad. Yeah, but today I met the maker of Chelsea. Sorry, I'm very excited. And so we could make this 10,000 maps, no? And with these 10,000 maps, we are okay accurate. So we're talking about 77% true skill statistic, which is not bad. Of course, it goes down when you validate a new species. That's normal. But accurate enough for what we have to do. We don't need to be pixel perfect, but we need to get the trends right. And when we get the trends right, then we can, make a pile of all our 10,000 maps and then have our number of species map for this 10,000 species of tree. Of course, it's not the 73,000, but we have 10,000 maps of 10,000 species all over the world, and this is pretty good. Okay, nice. Then we use this to study a number of things, especially about the ranges of the different species. But the thing I really wanted to do was to answer this third question. No? It's like, where will they be? Okay, and for this we had to run some climatic models. Okay, so we run the climatic models for the SSP 581, which is not business as usual, but a bit worse. I mean, nowadays maybe it's business as usual. And the good thing here is that we were in the original model. So this one, if you allow me to go back. So we were using only environmental covariates. Okay, so we're not using remote sensing data. Sorry, Fabian. But why we were doing that? It's because then the model was depending only on variables that could be estimated by the climate models. Okay, so by running the climate models, we had the inputs to then run predictions for each species, but in the future. Okay, we will take the globe, run the climate model, we get the temperature in 2100, and this is the input of our SDM. And by doing that, we get 10,000 new maps, but not of today, but in 100 years. I don't show you the maps because then everybody's like, I don't think in this place it can be like that. You remember, big uncertainty. So the only thing I show you is a kind of a summary where you can see basically who is winning and who is losing. I mean, unsurprisingly, there are lost species everywhere, but with a kind of a winner, which is boreal biomes. Okay, in boreal biomes, it seems that they will be able to profit from these new climatic conditions to basically thrive a little bit. But species will basically try to move in latitude and in altitude to escape the rising temperature. So the trends are kind of expected, but we could see them globally. Okay, oof. Are you okay? I'm still okay. Great. Now, of course, this is just a part of the equation. And I wanted to show you this first study just to show you what we can do now with this type of technology. So it's something that maybe you have already done on SDM, but at scale, more species, more resolution, and things like that. Now I want to attack a second problem, which is a problem that was beautifully raised in the previous talk, is that we have many of these inputs. Okay, we have many different variables: climate, soils, remote sensing, topography, location. We have texts. We have many things. Why do we have to choose? Can't we imagine a methodological framework when we don't have to choose, but we can use everything at the same time? And this is what I wanted to show you. It's called MaskSDM. It's not under review anymore. It's even published in Methods in Ecology and Evolution, but that was yesterday, so still I'm not too guilty, just a little bit. Okay, so the idea here is that we have all these different types of predictors. Okay, they come from different measuring systems. They have different strengths and different weaknesses. They show you different things. Okay, and now the story is that not only they are different, but they also come in different form. Okay, some of them are images, some of them are time series, some of them are also grid-like structure. Then if you can to imagine text, then it's words. What do you do with words? So let's assume that we are able to convert every one of these into some kind of vector form. And knowing that we have all this data, what are the things that you want your DeepSDM to be able to do? Come on, many of you work with SDM. Philip, you cannot answer. Or you can if you want, but you need to do it in the right order. Multi-species. Okay, multi-species, no problem, but it's not what I'm fishing for. Small scale, but why? We want big scale. Yeah. Oh, yeah. Okay. This again. This I can give you. Sorry. Wonderful. This is beautiful, but it's not on my slide. You're a great public, because usually people say exactly what they want to hear. But these are three things that are great and not in my slide, so please allow me to continue. So the first thing that you want your SDM to be, that's very much more mundane, actually, of the things that you have said. So the first one is that you want to be able to learn when variables are missing. This is one thing. If you create a model that needs temperature, soils, and satellite data from Sentinel-2, the day it's cloudy, you don't have Sentinel-2, you cannot run your model. I mean, we have ways to do it, but yeah. So, or maybe, you want a model that it's able to learn in absence of predictors. The second one, that almost multi-species, is that, different species have different needs. So for some species, you need to rely more on temperature and precipitations. Another, maybe it's more about proximity to human activities. And so you want to be flexible at inference time, so at prediction time, to the type of inputs that you have and be able to rank them. Okay? Because if you rank them, then you get the holy grail of AI and ecology, which is, come on. Sorry? Thank you very much. You already saw the talk. Yes, interpretation. So it's at least being able to tell me for these species, the interesting variables are this and this and that, and not this one. Okay? So MaskSDM, which is this model that we have been developing with sweat and blood in the last 2 years, tries to put these three things together. Okay? So it's a machine learning model, but a little bit more of the advanced type. Okay, we're talking about neural networks here. The principle is the same. You get some inputs, you try to predict the output. Every time you make a wrong prediction, oh no, 5. Every time you make a wrong prediction, you change the parameters until you get it right. Now, if you want to learn with missing records, so let's say sometimes a variable is missing, sometimes you have it, sometimes you don't have it. Usually what you do is that you don't use the modality. Okay, you say, sometimes I don't have the temperature one. Okay, temperature is not one of my predictors, because otherwise I cannot run my study and write my paper. Okay, or if you have it most of the time, you say, I do imputation, I take the average, before and after, and that's it. Here, we use a different strategy, which is exactly the same strategy that ChatGPT uses to learn. Okay, so this comes from text, and I don't know, do you use ChatGPT? You shouldn't, but if you do, basically, no, you're great, even if you do. So the story is that a language model is basically always trying to predict the next word. Okay, given the words I've seen before, try to predict the next. Now, how does it learn? Is that it masks some words sometimes, okay? So sometimes while learning, it will pretend not to know what this word is, try to predict it, and if it gets it right, no update of parameter. If it gets it wrong, update of parameter. If you do it 2, 3, 4 billion times, then you have a language model that you can use to do whatever you do with ChatGPT. So here we do the same, okay? So basically we start with all our, how can you be there, pointer? Okay, interesting. So you start with all these variables at training time, and you decide that, okay, this one I don't have. And in your model, you will mask it. Okay, you will pretend that you don't have it. And you will do it like statistically with a certain probability, but you will continue predicting your species with this model by assuming that you don't have some of the predictors. And thanks to this, you make the model robust to these missing variables. Okay, so that sometimes when you predict, and here is the prediction time, when you really don't have it, like in this case, it was a cloudy day, you don't have the satellite data, well, the model will just replace the vector it should learn with this masked vector that it has learned before, and it will still be able to predict. So it's a way to learn when you have only partial observations, and it's very robust to the fact that you are missing some of the variables. So we tried that with this data set called splot open, and so we have a 90,000 plot and 10,000 species, so multi-species model, yeah. and to be fast, because I don't have much time, here are the results. So long story short, this line here, is the one that we like, the one before the last year, okay? All these results are done by a single model, which is MaskSDM, and that's been trained by turning on and off all these different types of data here, okay? Every column is a model that uses only one or a set of these predictors, okay? So the game is to be better than all the other models that are learned on these specific combinations and can process only that specific combination. While the model here just doesn't care, okay? At inference time, you can turn on and off whichever of these predictors, as we call them up there, and has always more or less the same accuracy. Of course, if you try to predict 10,000 species only with temperature, it doesn't work so well. But I guess everybody here understands why. Okay, and then since we have learned to predict in absence of modalities, then you can very, very quickly obtain interpretability. You can just try to predict by turning off the variables that you want, and then it gives you immediately its importance. Okay, so in that case, you are able to assess the accuracy of your model while using a variable or not. And of course, you get the best results when you use everything, like here in the middle. And then you can also see that environmental variable, temperature, precipitation, and so on, is what brings you the most. And then other variables within that set, you can also rank them in terms of their importance. And this is all done at zero computational cost. You just have to pass your samples in and turning on and off the variables. It works overall. It also works while mapping, and these are the distribution maps that we can see. So I don't want to give my back to anybody. Yeah. So this is what you can obtain, for example, when you use all your predictors and inference time. So this is when you have them all. But then if you're missing the satellite data, for example, well, you will lose a couple of %, but the map will look like that. If you use only the temperature, you can see that you're still doing 88 something, but the map is very bad. So it's also important to assess your distribution maps visually, because a number just tells you part of the truth and not all of it. Okay, so this is it. This is what I wanted to tell you. I hope I'm not too long. I'm okay. I got a thumbs up. Yay. So the thing I want that I hope that you appreciated is what species distribution models and AI or deep learning allow us to do. And we can do large-scale, multi-species mapping, and it is something that works nowadays relatively well. But there is a lot to do still. Interaction, for example, which was on your wish list, it's there. We are working on it, and hopefully in some time we will be able to show some of that as well. Before I stop, I just wanted to give a big thanks to the person who actually did most of the things that I very gladly present to you. One of them is even in the room, so thank you, Benny. And thank you very much, and I'm here for questions.
Thanks a lot, Devis, for this inspiring talk.
Thanks.
Questions? Who wants to go ahead? Yep.
Hi. Thanks, Devis. Great talk.
Thanks.
The deep SDM or the neural networks, if you do it for multi-species predictions, does the model learn anything from species co-occurrences as like HMSC does? So would you call it the joint species distribution model, or is it agnostic?
I don't think so. Something must be learned indirectly. So, like we have seen that the species that have very few observations profit much more than those that have a lot. So there is some information sharing, but I will not say that it learns co-occurrences because it's very indirect. But we're working on that. We're actually with Loic, who is second time I should not do that, who is not in the room. We're working on like predicting assemblage and then trying to enforce some kind of interaction graphs in the prediction, but it is still a work in progress.
That was very inspiring. I have a question about how you may predict multiple species, and how do you solve the very high possible combinations you may have, like if you consider animal interacting with plants, for example. Yeah, how do you solve that? And is there any way to penalize the way that you get something by chance?
Okay, it's a very connected question. Yeah. So how do you predict multiple species? This is when using a neural network is quite straightforward. If you just need to change your loss function, which is the error function at the very end. So usually you try to pick one class by maximizing its probability and pushing down all the others. But if you replace that with something that does it species by species, one by one, then you're able to consider every species as a separate binary classification problem. Then your model becomes naturally multi-species. So this is the good news. The bad news is that, same answer as before, there is nothing explicit that enforces an interaction that will be biologically observed or known a priori, unless you encode it specifically in the model. But this is something we are really working on right now. I will say 99% of deep SDM in the bees now are just modifications of the architecture without any ecological knowledge a priori. But it's coming. Everybody, many people are interested by that, and it's coming. Give us 2 years.
Other questions?
No, come on. There is people on Zoom.
Thank you very much. Yeah, I want to ask, so it's actually related to the co-occurrence. If we add a few species as predictors, would that also be possible to integrate to the like the model architectures?
If you add species as predictors.
Yes, for example, just like as a vector instead of.
I mean, why not? I think there's a work by César Leblanc and Alexis Joly in Montpellier, who just came out in what was that, Nature Plants, I think. And that's more or less what they're doing. They are entering a list of species as text and let a language model predict the rest of the assemblage. So there are researchers going in that direction. I think it makes sense. Yeah, but you need something recurrent, otherwise. And then the big question is like, where do you start? No, which species you start in order to? Yeah, I don't know.
Robert.
Thanks for the interesting presentation. My question, you spoke about the future, and then my question is that many in the field of SDMs appearing many activities on generating some sort of standard variables, which appears as a black boxes, but can be used for modeling. I mean, for example, as you use the Google Earth engine, there is this embedding fields, which is 64 component data sets. And we don't know much about the insights behind, but it's, according to some studies, it is very good for predictions. So what's your opinion about this, and maybe
controversial opinions. Yeah, you're talking about Alpha Earth of Google, but I mean, there are several around. I think LNAI also disclosed one called Almo Earth, and you know there are multiple around. So this is like the recent trend of doing what we call a foundation model, right? And this foundation model will be, let's take all images on Earth, put them into a black box, extract some vectors, and these vectors will represent that place. And then you can use them for whatever you want. I mean, it's a different approach of what we're talking about here, which is more about integrating ecological knowledge into models, but maybe one doesn't exclude the other. If you look at a little bit in detail here, but I really didn't talk a lot about it, there is this sat clip here, which is one of these generic features. And we use that instead of using the satellite images. So we didn't have to download the satellite images. So, but supposedly those features represent that location and have been learned on a gazillion Sentinel-2 scenes. So in theory, that vector should represent what we usually see in satellite images. Then it's I guess it's mostly a matter of faith than anything, but I mean, they exist. Why not give it a try? But I will not trade our ecological curiosity and knowledge with that. I will synergize rather than replace.
Thank you very much for the lively discussion. We are at the end. We have still more questions, and I think, yes, there is more time to connect to the speakers, but I hand over to Catherine. Thanks a lot, Devis.
Thank you.
So as you know, we've almost come to the second part of our program, but we'd first like